Hi everyone and welcome to the microbiology section of First Aid Express. My name is Sean Alemi. I'll be guiding you through 4 sections of microbiology. We'll start off with general information about the classification of bacteria, and then we're going to jump into specific information about individual bacteria, fungi, and parasites. Over the next few lectures, I'm going to present information mainly from microbiology, but I'm also going to take every opportunity to create integrations for you. I'll pull in pathology, neuro, pharm, physio, as much as I can to help you create those unforgettable connections. Along the way, I'm going to ask you a lot of questions. Some of them will be rapid fire, others will allow you more time to think. Don't be discouraged if you don't know the answer. The point of these questions is to help you learn by exposing what you don't know. I'd rather that you miss it here with me and get it right on the test. Okay, so enough with the formalities, let's get started. Next up is clinical bacteriology. Let's do it. In your book, you'll find a great flow diagram for working up gram-positive organisms. The key thing to remember here is that the gram stain is always your first step. Once you know if the bug is gram-positive, then you can start asking questions about the characteristic of that bug. I think the diagram will actually make more sense and be easier to tackle once we have covered all of the gram-positive organisms. So by the end of this lecture, hopefully you'll be able to come back to it and know all the information presented here. But the thing I want you to take away is that there are always a few key features you must know about every bug. First, the shape. Are they cocci or bacilli? Or are they completely different and in the shape of branching filaments? After you figure this out, then you can start looking at enzymatic activity, things like catalase or coagulase, or perhaps the bug is hemolytic. These are all features that we will discuss as we go through the bugs individually. While we were on this algorithm, let's do a quick exercise. Let's say that you gram-stained an unknown bacterial sample and found that the color of the bacteria was purplish blue. This tells you that it is a gram-positive organism. A quick word on shape of the organism. Gram-positive organisms will have one of 3 main shapes: cocci, which you can see here, or bacilli, which you can see here. Or they can have branching filaments, which you can see here. Let's say that we see a purple-blue cocci-shaped gram-positive organism. This means that there are only 2 possibilities for the organism that we are dealing with: Staphylococcus or Streptococcus. It's easy to differentiate the two. Staphylococcus will present in clusters and will produce catalase. Streptococcus will present in chains and will not produce catalase. Catalase is an enzyme that breaks down hydrogen peroxide into water and oxygen. Just remember that staph will make catalase because it has the extra staph to do so. So, you see purple-blue cocci in clusters that are catalase positive. You've identified that it is Staphylococcus. The next enzyme we're going to assay for is coagulase. Coagulase is an enzyme produced by Staph aureus which enables the conversion of fibrinogen to fibrin and results in the clotting of blood. Coagulase-negative staph that do not clot blood will be Staph epidermidis or Staph saprophyticus. So what if the organism was purple or blue cocci in chains that are catalase negative? What have you identified? You've identified Streptococcus. But you still need to determine which of the several important strep species you have, and this is done by looking at the hemolysis patterns. Hemolysis is the breakdown of red blood cells in culture by hemolytic enzymes found in some strep species. There are 3 main types of hemolysis. Alpha hemolysis is partial hemolysis that results in a green ring pattern around the strep pneumonia or viridans streptococci in blood agar. The green is from the color change in agar due to peroxide produced by the bacteria. Beta hemolysis results in complete clearing around group A or group B strep. This is due to hemolysin produced, which completely lyses blood cells. Gamma hemolysis results in no clearing or hemolysis because hemolysin is not present. Make sure you go back and review these. These key enzymes are highly likely to show up in a test question. There are a few antibiotics that can help us in identifying gram-positive cocci. For differentiating staph, Novabiocin is helpful because saprophyticus is resistant and epidermidis is sensitive. Saprophyticus resistant, epidermidis sensitive. Remember this association with the following mnemonic. At the staff retreat, there was no stress. Bacitracin and opticin are two antibiotics used to identify strains of streptococci. Opticin is a chemical that is useful for distinguishing strep pneumoniae and viridans strep. Specifically, viridans is resistant and pneumoniae is sensitive. Viridans resistant, pneumoniae sensitive. Bacitracin can help differentiate group A from group B streptococci. Group B is resistant and group A is sensitive. Group B is resistant, group A is sensitive. You can remember these associations with some cool mnemonics. For opticin, remember overpass, with the first letter of each word forming the mnemonic. And for bacitracin, just remember B bras. Remember that alpha hemolysis is an incomplete hemolysis, causing a greenish ring around the colonies. The two streptococcal chains that cause alpha hemolysis are strep pneumo and viridans strep. When trying to differentiate the different streptococci species, those that produce beta hemolysis or complete hemolysis when grown in blood agar are which two bacterial species? I'm thinking of group A strep, like strep pyogenes, and group B strep, like strep agalactiae. You should also know, however, that there are two other gram-positive organisms that have hemolysin and can demonstrate beta hemolysis. These are Staph aureus and Listeria. Okay, time for our first flash quiz. This is a fill-in-the-blank question. Opticin, the antibiotic, can be used to differentiate between blank and blank. Fill in those two blanks for me. And the answer is that it helps differentiate between strep viridans and strep pneumo. Remember that strep viridans is resistant and strep pneumo is sensitive. You can use the overpass mnemonic.